Tricarboxylic acid cycle. Introduction. The German-born British biochemist Sir Hans Adolf Krebs proposed this cycle in 1937. Tricarboxylic acid cycle, TCA cycle, also called Krebs cycle and citric acid cycle, plays a central role and is the second stage of cellular respiration process by which living cells break down organic fuel molecules, that is glucose and some other sugars, fatty acids and some amino acids in the presence of oxygen to harvest the energy they need to grow and divide. These metabolic processes occur in most plants, animals, fungi, and many bacteria. In all organisms except bacteria, the TCA cycle is carried out in the matrix of mitochondria. Large molecules must be degraded before they can enter the TCA cycle. These are broken down into a 2-carbon compound called acetyl coenzyme A, acetyl CoA. Prior to the TCA cycle, glycolysis occurs which generates molecules including pyruvate, ATP and NADH. Pyruvate is then decarboxylated by the pyruvate decarboxylase complex to form acetyl CoA. It is the intermediate that enters the TCA cycle. The TCA cycle is a central pathway that provides a unifying point for many metabolites which feed in at various points. Though the primary role of the TCA cycle is production of NADH and FADH2, it also produces molecules that supply various biosynthetic processes. These enter or exit the cycle at various points depending on demand. The TCA cycle consists of 8 steps catalyzed by 8 different enzymes. Steps of the cycle. Step 1. The first step is a condensation step in which acetyl CoA, 2-carbon molecule, reacts with oxaloacetate, a 4-carbon molecule, to form citrate, which is a 6-carbon molecule. The enzyme involved in this step is citrate synthase. CoA is bound to sulfhydryl group SH and diffuses away to eventually combine with another acetyl group. This step is irreversible because it is highly exergonic. The rate of this reaction is controlled by negative feedback and the amount of ATP available. If ATP levels increase, the rate of this reaction decreases and vice versa. Step 2. This step involves 2 reactions. First, citrate loses one water molecule to form cis-aconitate and gains another water molecule to form isocitrate, an isomer of citrate. The enzyme involved in both these reactions is aconitase. Step 3. Isocitrate is oxidized to alpha-ketoglutarate, a 5-carbon molecule, which results in the release of carbon dioxide and 2 electrons, which reduce NAD+ to NADH. One NADH molecule is thus formed. The enzyme responsible for catalyzing this step is isocitrate dehydrogenase. This step is also regulated by negative feedback from ATP and NADH and by a positive effect of ADP. This is a rate-limiting step as isocitrate dehydrogenase is an allosterically controlled enzyme. Step 4. Alpha-ketoglutarate is oxidized to form a succinyl group, a 4-carbon molecule. This molecule binds to coenzyme A, forming succinyl CoA. A molecule of NADH and a molecule of carbon dioxide are produced. The enzyme involved in this step is alpha-ketoglutarate dehydrogenase. This enzyme is regulated by the feedback inhibition of succinyl CoA. Step 5. A phosphate group is substituted for coenzyme A in succinyl CoA and a high energy bond is formed. This energy is used in the substrate level phosphorylation to form either guanine triphosphate GTP or ATP. Succinyl CoA is then converted to succinate, a 4-carbon molecule. The enzyme involved in this step is succinyl CoA synthetase. There are two forms of the enzyme depending upon the type of animal tissue in which they are found. One form is found in tissues that use large amounts of ATP, such as heart and skeletal muscle. This form produces ATP. The second form of the enzyme is found in tissues that have a higher number of anabolic pathways, such as liver. This form produces GTP. Thus, the result is one GTP molecule is produced. GTP is energetically equivalent to ATP, however, its use is more restricted. Step 6. Step 6 is a dehydration process in which succinate is converted into fumarate, a 4-carbon molecule. Two hydrogen atoms are transferred to FAD producing FADH2. This carrier remains attached to the enzyme and transfers the electrons to the electron transport chain directly. This process is made possible by the localization of enzyme catalyzing this step, which is succinate dehydrogenase, inside the inner membrane of the mitochondrion. Step 7. Water is added to fumarate, which converts fumarate into malate, another 4-carbon molecule. The enzyme involved in this step is fumarase. Step 8. This is the last step in the citric acid cycle in which malate is converted into oxaloacetate. The enzyme malate dehydrogenase regenerates oxaloacetate by oxidizing malate. A molecule of NADH is produced. By regenerating oxaloacetate, this cycle continues. Products of the citric acid cycle. 2 carbon atoms come into the citric acid cycle from each acetyl group, representing 4 out of the 6 carbons of one glucose molecule. 2 carbon dioxide molecules are released on each turn of the cycle. The 2 acetyl carbon atoms will eventually be released on later turns of the cycle. Thus, all 6 carbon atoms from the original glucose molecule are eventually incorporated into carbon dioxide. Each turn of the cycle forms 3 NADH molecules and 1 FADH2 molecule. These carriers will connect with the last portion of aerobic respiration to produce ATP molecules. 1 GTP or ATP is made in each cycle. Several of the intermediate compounds in the citric acid cycle can be used in synthesizing non-essential amino acids. Therefore, the cycle is amphibolic, which means it is both catabolic and anabolic.